Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And you can always find Let's Talk Micro on Apple Podcasts, Spotify, Google Podcasts, Amazon Music, TuneIn Radio, Good Pods, wherever you listen to podcasts, you can find Let's Talk Micro. As far as social media, I am on TikTok, YouTube, and Instagram as Let's Talk Micro, on X as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and I have an email address which is letstalkmicro@outlook.com. So either via social media or via email, you can send any feedback, any topic suggestions. They are always welcome and appreciated. So please go ahead and subscribe to the podcast, download episodes, and if the app allows you to do so, please go ahead and leave a review. It always helps the podcast, and I am always grateful for your support. If you haven't checked out the previous episode, please go ahead and do so. It was part 3 of the AMR sub-series, so the third episode in sub-series. So as you know, if you've been following the podcast, the first episode was AMR, an overview. The second episode was about beta-lactams. And the third one, which is the last one, was about fluoroquinolones. And the first episode of the sub-series was episode 98, and that was the overview. And then the second episode was episode 101, which is the one where Dr. Brian Rowe, he came into the podcast and talked about beta-lactams. So definitely check them out if you haven't already. And if maybe this is the first episode you're listening to within the podcast, where I talk about organisms, reactions, I bring guests. I try to explain terms as simple as I can. We created a sub-series which is about antimicrobial resistance, in which Dr. Andrea Princey, she's the co-host of this series with me. And then we just really break down antimicrobials. So I think it's very educational. It is a way of bringing all this information that we need to know as part of our jobs, but it can be very daunting, very challenging. So on the previous episode, Dr. Minky Wongwatana, which he's a pharmacist that works in the industry with a lot of experience, he joined the podcast on that episode to talk about fluoroquinolones. So he gave us a historical overview. He talked about the mechanism of action. He talked about the breakpoints for salmonella and fluoroquinolones. A lot of great information, all in all. And it was wonderfully explained by him. And those of you that are listening, so as we learn about drugs, we know that they have different mechanisms of action. And we know with the beta-lactams is, that's the target is the cell wall. And in this case, in the case of fluoroquinolones, is DNA. So definitely great information to know. And I hope that if you haven't checked it out, go ahead and do so. But Minky explained it so great, so thank you. So most definitely learning about antibiotics, it's a lot of information, a lot of knowledge. It can be challenging, it can be daunting. So if you're looking to learn more, I invite you to go ahead and check out learnantibiotics.com and also check out the book, which is Learn Antibiotics, which is available in Amazon. And it includes, cheat sheets, practice tests, games, and more. And these resources, they are being used by thousands of people worldwide and may be helpful. So I invite you to go ahead and check them out. So today's episode is about Streptococcus pneumoniae. Those of you that have been following the podcast, you know that a couple weeks ago, episode 119 came out, which was about Streptococcus pneumoniae. And this was an episode that was originally published as episode 57. And in it, I go over Streptococcus pneumoniae. I talk about the Gram stain. I talk about morphology. I talk about uptaking and bile solubility testing. And there was an episode that followed that one originally, which was episode 58, which was about Streptococcus pneumoniae testing. So this episode, it's that original episode, but with some changes. But it is about testing on Streptococcus pneumoniae. And I talked about urine antigen testing, I talk about molecular testing, and I also talk about the importance of correlating those results that you get from that type of molecular testing with the Gram stain and morphology. All in all, a great episode, and for the benefit of new listeners and as a way to kind of go back and refresh on some content, refresh our knowledge, please go ahead and enjoy this episode about Streptococcus pneumoniae testing. Let's go ahead and listen to it. So now that I have covered morphology, biochemicals for strep pneumo, let's talk about other ways of identifying it. let's talk about other tests. Well, those of you that work in a lab, you know that you might have seen it. There is a popular antigen test called the Binax Streptococcus pneumoniae antigen card, and this is made by Abbott. This is a rapid immunochromatographic assay for the detection of Streptococcus pneumoniae on patients with pneumococcal pneumonia and meningitis. The test detects pneumococcal soluble antigen in human urine and CSF, and this is from the package insert of the test. The card has control antibody and rabid anti-strep pneumo antibody. And what kind of samples are used? Well, for pneumonia, you need urine, and CSF for meningitis. I have never seen it perform in CSF in the lab. I mean, it's always for urine. So the test comes with the test cards. It has a reagent A, which contains citrate and phosphate buffer. It comes with specimen swabs and controls, which are a positive and a negative swab. And these are color coded. So you have a red swab, which is your positive control, and you have a blue swab, which is your negative control. So the positive swab has inactivated strep pneumo antigen, and the negative swab does not. So let's go ahead and start with specimen requirements. So you need urine that has been stored at room temperature for 24 hours, or refrigerated or frozen up to 14 days is acceptable. Boric acid preserved urine is also acceptable. So those of you that work in the lab, you see the little gray tops where you put urine that contains boric acid, and that's acceptable. So if you get, so if you're missing a test, you can use one that came from your analysis. So CSF stored at room temperature for 24 hours, or also refrigerated is acceptable, or samples that have been frozen for up to 1 week. And when I mention urines, if they have been frozen up to 14 days, that's acceptable. So how is the test performed? Well, you bring the samples to room temperature. Then your test card has 2 holes. You did the swab in the specimen, and they place it in the bottom hole, and you push it upwards. Then you add 3 drops of reagent A to the bottom hole. The inside of the card has an adhesive liner. You peel that off and you close the card. Once you close it, you can leave it at room temperature and read after 15 minutes. And if you are testing controls, you add 6 drops instead of 3 to the bottom of the hole. So what does a negative and a positive look like? So a negative result is a single pink to purple color line
in the top half of the window.
This line means that the test was performed correctly. And then a positive result is 2 lines. You have your control line and a pink sample line. The intensity of the line depends on the amount of antigen present in the sample. Any line for the test is a positive line. And I like to stress that out. Any line, any intensity of a line, it's a positive result. That's something that I always like to mention. And those of you that work in the lab can relate to this, which is, right? Sometimes, you perform your test and you see the line, maybe very faint. You put it against the light and you're looking at it. You ask a coworker, do you see a line here? Do you don't see it? Are you a little afraid to report it? Maybe repeat it. Barely see the line. Do I call it a positive or a negative? So this test, and this is not only with strep pneumo, other, chromatographic tests, and that you have, a test line and a control line. So typically any intensity of a line, so any line, regardless of intensity, is a positive result. So this is something to keep in mind. Whether it is a very strong line or very faint, it is considered a positive result. And just like other tests where you have a control line, so if you have a test where you don't have any lines, or your control line is missing, this is considered an invalid result. And at that point in time, you need to repeat the test. So always make sure, like I mentioned, this is a test that you add the drops to the swab, and then you close the card, and then you wait 15 minutes. So this test definitely has walk away capabilities. You can go ahead, set them up, set a timer, and go ahead and continue doing another test. But what you have to make sure is that when you close the card, you make sure that the card is closed properly. Because a lot of times, if you don't do that, maybe you were light when you closed it, or it can just pop open. So just make sure that it's properly closed before you walk away. And something else before you walk away, make sure that the actual, that the sample is migrating. Because, you put your swab, you add your drops, you close your card and make sure that the sample starts migrating up. Because sometimes it happens, like you go ahead and you do your testing, you close the card, set your timer, walk away, and then when you come back in 15 minutes, you realize that it hasn't migrated. So then you have to go ahead and repeat the test. So in the lab, sometimes, it's all about time, making sure we perform our test correctly. So you need to make sure those two things before you walk away, so you don't waste time, which are make sure it has migrated and that the card is properly closed. As far as limitations of the test, it was validated on urine and CSF samples. So other samples were not validated. It has a 90% specificity. And having said that, Bailey and Scott's Microbiology, a great textbook that I use constantly, says that urine antigen testing has been proven effective for adult patients that receive antimicrobial treatment prior to a primary culture. So meaning that before a culture was performed, they had antibiotic treatment, and that's when the test, performs the best. So it does not differentiate between present and past infections. And according to the American Society of Microbiology, for their Manual of Clinical Microbiology, this test is positive in patients that are carriers without infection. So remember that I said before that you can carry this organism without it causing any problems, and you can have a positive test. And this is actually often observed among children. So it should not be used in children below 6. And it is recommended in adults that when you perform this test, you perform it along with a traditional culture. So just not by itself. You should be doing some sort of culture, like a sputum culture or something like that, when you are performing this test. So definitely keep that in mind when this test has been performed. Has good specificity, but it cannot differentiate between a past and a current infection. And carriers might have a positive test for this. But it's a very popular test. You definitely see it a lot in the micro lab. And those of you that remember, we definitely saw an increase with COVID. They were ordering sputum cultures, Legionella, Strep pneumo, and sometimes, there were shortages and they were back order. So definitely, we struggled with this during COVID as far as having proper supply, numbers. And like I mentioned before, since Streptococcus pneumoniae can causes meningitis, it is part of your meningitis panel. So when those of you that work in the lab and you run a meningitis test, for example, like a very popular one is from BioMero. They have the BioFire system, and there's the ME panel, the meningitis panel. So Strep pneumo is one of the targets. For you, the audience, what other organisms would you expect to see in a test like this? I'll give you a few seconds to think. Well, you definitely see E. coli. You see Streptococcus agalactiae. You see Cryptococcus neoformans. So, these organisms, they cause meningitis, so they are part of this meningitis panels. So this is from the BioFire, the ME panel. So you also have, as far as blood cultures, you have, as part of the PCR molecular nucleic acid testing, Streptococcus pneumoniae is one of the targets. For example, Luminex has a very gene system for blood cultures that has a gram-positive panel for blood culture samples. So one of the targets is Streptococcus pneumoniae. And this is where that gram-stain morphology comes in handy. Remember, I've been saying so far, it is a gram-positive coccyne pairs. It's a very distinctive morphology, and you as a tech, you as a student, I've been asking you to keep it in mind, to remember it. And this is why. With this particular panel, one of the limitations of the system is that Streptococcus mitis can have cross-reactivity with the strep pneumo probes. So what does this mean? It means that maybe the sample has Streptococcus mitis, which is another alpha hemolytic strep. The test can yield a false positive result for strep pneumo, and I have seen it. So typically when you are working blood cultures, right, your instrument flags the bottle as positive, you go ahead and do your gram stain, and you go ahead and report that. And then if you have one of the setups that where you can run a molecular test, you go ahead and run your test. It typically takes about 2 hours, and then you go ahead and update your result. So, for example, you have gram-positive coccyne chains, you call it to the floor, and then your result comes back as strep pneumo from that molecular test. And then you go ahead and update it to strep pneumo, and then guess what? It might not grow on the plate. You might get strep mitis. When I talk about remembering that morphology, if you are running this particular test and you get a strep pneumo result, correlate that with the gram stain. Maybe if you saw regular gram-positive coccyne chains, I would recommend holding off on the result, document it internally, and then verify the ID once the organism grows on the plate. But if you get the strep pneumo ID, and then you go to your gram stain and you remember that it was gram-positive coccyne pairs, then it will be okay to release that result. So just be careful. Granted, like I mentioned before, organisms, they do not go to school, they don't read books, so they do whatever they want. There are many factors, like antibiotics and other things that can affect the morphology of a cell. So you can sometimes have atypical presentations, and you could have a strep pneumo that just doesn't look, it's not the gram-positive coccyne pairs. But, it happens. You can have unusual presentations. But what I'm asking is, just if the gram stain doesn't correlate with that ID, hold off on reporting it. Just document it internally and then verify the organism once it grows on the plate. And like I said, you can have atypical morphologies. I once had a listeria that this varying system called listeria, and the gram stain looked nothing like listeria. It wasn't, it wasn't the short gram-positive rods. So we, held on reporting that ID. It was documented internally, but guess what? When it grew on the plate, it was a listeria. So you do have atypical presentations, but you always proceed with caution. That way you don't make an error and erroneously report something. As far as other systems, BioFire, again, it also has a blood culture panel called the BCID2 that it also identifies Streptococcus pneumoniae. But it does not seem to have that cross-reactivity issue. So there are quite a few tests out there that can identify strep pneumo. There are some things that you should keep in mind. Now with MALDI-TOF, you might be seeing more of another member of the S. mitis group, which is called Streptococcus pseudopneumoniae. Because if you look at once you're looking at your textbook, strep pneumo, it's part of the strep mitis group. So this organism, Streptococcus pseudopneumoniae, it was separated from strep pneumo. It causes respiratory tract infections in patients with predisposing conditions such as COPD, which is chronic obstructive pulmonary disease. So it is not encapsulated. It is insoluble in bile. So remember the bile solubility test. So it is insoluble, so it will not lyse. So if you have suspension, it should remain turbid. And this organism, it is susceptible to optogen only when it is incubated in non-CO2. And as far as optogen, You can also have some strains of Streptococcus oralis and Streptococcus mitis that can be susceptible to opticin. However, they are insoluble in bile. So once again, insoluble in bile means that there will be no ghost colonies or clearing of the deoxycholate solution. So if you're doing the colonies, there will be no clearing because there's no lysis. And if you're doing the tube, the suspension will remain turbid. So keep that in mind when you're working in the bench, when you're reading opticin disc, when you're performing IDs. So if you get this organism, Streptococcus pseudopneumoniae, it is insoluble in bile, so it has a negative bile solubility test, and it is non-encapsulated. And only when it's incubated non-CO2, it can be susceptible to opticin. So keep that in mind. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about Streptococcus pneumoniae testing. As always, I enjoy sharing this information with you. And definitely I invite you to check out that previous episode about Streptococcus pneumoniae, episode 119, where I go over morphology, I go over opticin, and more. As always, I am grateful for the support. Thank you for downloading episodes. Thank you for following on social media. Please continue recommending Let's Talk Micro to your coworkers, to your students. I enjoy doing this so much. As always, continue bringing that passion to what you do. It's so important. You do such great work. So as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.